much. We say thank you very much for finding out time to be with us in this training. Most of us have to squeeze out the little we have just to ensure that we participate in this training. I want to say thank you. Why I'm thanking you is not for my own good, but it's for your own good. So can you imagine with telling you thank you for your own good? Yes. So we know it is not easy. So we are saying thank you. And also I want to say a very special thank you to please mute your mic. So I want to say a very special thank you to some set of persons. Okay, I'm coming. I want to say a very special thank you to some set of persons, especially those from Uniben. The registration from Uniben was massive, and that appreciation goes to Professor, let me call him Professor V.O. Emeje. I want to really say thank you for that. The registration from Uniben was so massive. So I owe Uniben a gift. Yes, we owe Uniben a gift. So Uniben, get ready. yeah, we owe you a gift. So thank you so much. Okay. And also to other schools, Michael University of Agriculture, people did well, and to those who shared our adverts, we are saying thank you. Thank you so much. So a quick one, we are going to start.
Hello?
We are going to start.
Yeah. Hello. Hello.
So
sorry, I was in the hospital with someone.
Mute your mic.
No, well, I took some students there to visit patients.
Mute your mic, please.
Mute your mic.
Well,
So
they've been on strike.
Let me unmute myself. All right. So to avoid that stress of me coming to unmute you, coming to meet you, so it's better you just do that by yourself, so the class can be smooth. I'll quickly share my screen right away. Right. Are you seeing my screen now?
We can see it.
Yes,
Okay.
we can see.
Thank you. So once again, on behalf of Hendeb Industries Nigeria Limited, I, Osiago Henry Chibueze, we welcome you. I will be your facilitator for this training. The training is being anchored by School of Computational Chemistry and Biology. That is the training division of the company. So from now to the one more time, we shall be talking about Pharmacognosy and in silico drug design. Based on the statistics of those who registered, a good percentage of the participants are in the area of chemistry. A good percentage. So that means the training is for you. Why some persons are from pharmacy, pure pharmacy, then microbiology. So we shall be looking at what the training has for us. We'll look at the course module. We'll look at what we are going to do, the things we are supposed to achieve, the areas we are supposed to cover from now till the end of the training, what we are expecting in this training. So these are what we'll be looking at. And then we shall look at our module.
You know what? Can you come out of
Our
this?
module.
Yeah. I should
Uh,
press that.
so now I will start by saying this. In India, 80% of Indian relies on their traditional medicine. A huge number of Indian citizens relies mostly on their traditional medicine. Same is being applied to China. They are currently encouraging traditional medicine. In fact, I had a training yesterday with Genomark Institutes. Some of us are familiar with Genomark Institutes. I was privileged to facilitate the training for them on drug design. We wrapped up that training yesterday. And one of the participants, Dr. Bini Nathaniel, he made a crucial comment that when he traveled to India for treatment, the hospital where he was administered, when they wanted to prescribe some drugs for him, they gave him two options. Do you want us to give you traditional medicine or conventional medicine? In their pharmacy, they sell both traditional medicine and conventional drugs. So he was like, this option, okay, let me go for both of them. But he opted for traditional medicine. So he was treated with those traditional medicine. He testified yesterday that the traditional medicine was much more effective than the conventional drugs. That he even had to come back to Nigeria with some of those traditional medicine. So if you visit pharmacy shops in India, they sell these herbal products. The long reach product we use in Nigeria are herbal products. So that is to say that there is a future. There is a future for us in pharmacognosy. In Nigeria, traditional medicine is still, in fact, has not been explored until we begin to see herbal products being sold in our pharmacy shops. Then at that point, we know that we are doing something in traditional medicine. But up to this point, in fact, we barely have this perspective to even patronize traditional medicine. So the essence of this training is to see how we can turn our plants to medicine. Now, we don't just train. In Hendeb, we don't just train, but our aim of all our training is to see how our participants can exploit these tools and be able to develop a prototype. Even if they cannot develop a product, because you need facilities to develop a product, but you don't need much facilities to develop a prototype, a laboratory prototype that has been tested and is effective. So this is the essence, the whole essence of this training, to expose us into the steps involved in turning our plants to medicine. Last week, Wednesday, I traveled to some parts in Nigeria. While I was on transit, I was just looking at our forestry. I saw that we are blessed with lots of medicinal plants. So once I came back, I began to call some of our herbalists and began to see how we can work together. So we are patronizing some herbalists. We want to work with them because they have this knowledge, but they lack the knowledge of converting what they have into a scalable product. So we are blessed in Nigeria. So the training is timely. It's timely, and we'll do justice to that. So by way of introduction, the training overview. The 1-month certificate course in Pharmacognosy and in silico drug design is an intensive, skill-driven virtual training designed to bridge the gap between natural product research and modern computational drug discovery. Just like our slogan is, bridging the gap, reaching the world. There is a gap. There is a gap between natural product research and the modern computational drug discovery. We know that it is very expensive to come up with synthetic drugs. We don't have the facilities for synthesis. We don't even have the facilities for isolation. But I believe that we have the facilities that can tabulate these herbal products. You can even have them in tea form. The problem we have been having is people have been complaining of the issue of dosage and standardization. At the end of this training, we'll be able to see that those things are not really our problem. They're not really our problem. So this training will bridge that gap between natural product research and computational drug discovery. This program will provide participants with hands-on exposure. So it will not be a theoretical training. It will be a training where our hands will be on our keyboard. So we shall expose you to contemporary tools used in drug discovery. If you look at the process involved in drug discovery, before in silico tools came up, it was expensive and time consuming. But now we have some of these contemporary tools that has eased up. And in fact, they call it computer-aided drug design, meaning computer tools that aid drug design. So we shall expose you to those contemporary tools. We shall expose you to molecular modeling. We expose you to pharmacokinetics, toxicology, and bioinformatics, with a strong emphasis on natural products. So all what we shall be discussing lies on natural products and phytochemicals. The aim of this training, the primary aim of this program, is to train participants on how to identify, screen, and evaluate bioactive compounds from natural sources. So we shall learn how can we identify bioactive compounds. We'll learn how to screen bioactive compounds and how we can evaluate their efficacy. All this is expensive and time consuming if you want to do them in bench work, if you want to do them in wet lab. But we shall now see how we can achieve them using computational tools. We shall learn how to integrate pharmacognosy principles with in silico drug design approaches. So we'll see how we can be able to integrate both of them together. The training will equip our researchers and students with industry-relevant computational skills. Remember that our aim is not just to train you, but to enable you to develop a prototype that you can then relate with the industry. I was speaking yesterday in the training and I told them that there is a weak link. You know, there's a triangle in research. There's a triangle in research, and that triangle in research have been tempered. We have our government. We have our government. We have our academias. Then we have our industry. We have our industry. This is the triangle in research. But then there is a weak link here. Here is weak. Here is weak. There is no connection again between academics and industry. Instead, what we do in the academics is to write papers and publish. Papers without products. And that is not what we are after. We are after developing a prototype, a laboratory scale, that, okay, we can now meet the industry and say, we have a product. Let's collaborate. Not we have a paper. Industry cannot collaborate with an institution that has lots of paper. I've never seen an industry that collaborated with an institution because the academics, their researchers publishes 1,000 paper in a month. We are not interested in your volume of paper, but we are interested in the product you have. So our aim is to provide our researchers and students in this training with industry-relevant computational skills for drug discovery, publication, and innovations. Now, we have 8 modules. So we are going to explain those 8 modules in this training, and so you can be able to see what you are expecting. One of the first areas we shall start, we should begin with, is Phytoinformatics. So we shall begin this training with Phytoinformatics. In Phytoinformatics, the participant will be introduced to reputable phytochemical and natural product database. So we shall expose you to databases of phytochemicals. I have about 5 of them where you can look out for natural products and phytochemicals. These databases are used in modern drug discovery. Now, the module will also cover the source of phytochemicals, be able to source for phytochemicals from medicinal plants. We'll learn how to retrieve compound structures and biological data. We'll be able to understand compound identifiers. Then we'll be able to prepare phytochemicals for computational analysis. Overall, participants will learn how to systematically mine phytochemicals associated with specific plants or disease. So we can mine phytochemicals based on disease. We also learn how to mine phytochemicals based on plants. Let's say you are working on diabetes, so you want to mine bioactive compounds capable of treating diabetes. So we shall look at how to mine that, so that you will not just be using anyhow compound to test for diabetes, but you now streamline your research to compounds that has the features of controlling diabetes, of interacting with the diabetic cells. So these are what we shall look at in phytoinformatics. And then we shall also look at chemoinformatics. That is another aspect of the training, chemoinformatics. So we shall look at for chemoinformatics. Now, what are we going to learn in chemoinformatics? So by the time we are done with phytoinformatics, we are moving ahead to chemoinformatics. And this session we focus on global chemical and small molecule database. So we have some chemical database which we're going to expose you. You learn how to retrieve chemical data. We shall be able to see how we can access chemical libraries. Because one thing about drug discovery is that you must be able to create your library. So we will teach the participant how to build their own library. For drug design, I have my library, I have my own database. So one of the our assignment will be how to create your own database. All right, are we together? All right.
Yes, we are together.
All right. We shall also learn. structure retrieval and optimization. My major interest at Techno-Informatics is structural optimization. How to optimize our structure to make them have those features that will enable effective interaction with our disease receptor or disease target. Then we shall learn compound filtering and standardization. Not all bioactive compounds are medicinal compounds. So we shall learn how to filter out those bioactive compounds that are not medicinal. Focus only on those ones that are medicinal. You may have a GCMS result from a plant, and imagine you are seeing phenol and you are seeing benzene among the GCMS. One who don't know compound filtering might decide to just dot everything, but definitely the benzene and phenol will not give you a good result because they lack the features, the drug-like features. So we learn how to filter our bioactive compounds and standardize them to the ones that has the features necessary to interact with our target. Then we shall understand the role of physiochemical properties of small molecules. There are some physical and chemical properties of our bioactive compounds that controls their interaction with our disease targets. We shall look at that in chemoinformatics. So this is another module. At the end, this knowledge forms the foundation of downstream computational screening. We have upstream, downstream, and midstream. So this forms the foundation for downstream computational screening. Another module that I sent this module to our email, to our drive. So it's already in our drive, so you can look at it at the end of the session. We should expose it to bioinformatics. Bioinformatics also is part of what we're going to learn in this training. So we are taking our time today, being the one to let you know what the training has for us. So we expose it to bioinformatics. Bioinformatics is too broad, so the part that has to do with Pharmacognosy, we shall expose it to those parts. Participants will be trained on bioinformatics resources essential for target identification and validation, including protein databases, structure and sequence based. So we shall be able to learn protein database, the ones for structure and the ones for sequence. Most times, you may end up not having the desired protein in any of those databases. But once you have the amino acid sequence, you can now model the protein. So it will be part of what I will introduce to you. Because not all disease target has been modeled and deposited in protein database. Yes, the only modeled 3D structures of protein, the only ones you can find are the ones that we are modeled by scientists. And we cannot claim that they have modeled all the protein. The diseases are emerging and re-emerging. So those re-emerging disease, the emerging ones that are new, they don't have proteins. I was doing a work on Ascaris lubricants. I hardly found a protein in the protein data bank. So there are some of the research you want to do. You search for that protein, they are not there. What do you do? So learn how to get their sequence and model them. Then we shall look at disease-related target identification. How can we be able to target the receptors, the genes that are associated to our disease? Shall expose you to this area. Definitely, a disease does not have a single interaction. Every disease have multi-interactions. So a disease have multi-interactions. And while you want to discover a drug for that disease, then you must look out for those possible mechanisms of that disease and get their target. You look out for those
I think
mechanisms.
so. Yes.
please mute your mic. So you
Sorry,
look at
remove it. I'm sorry.
those
Sorry.
possible mechanism of that disease and attack them. But most people just rely on one mechanism. Meanwhile, these mechanisms are interconnected. So you can build a network that shows the most connected mechanism. There are so many biological processes involved in a disease. That is why before we start working with a disease, we study the pathogenesis of that disease. That we unravel the mechanistic view, the mechanism of the disease. So we shall be able to now identify the related target of that disease. So that if you, I told somebody last week that if you want to do a docking on a disease, if I see a paper where they docked with 1 protein or 2, in fact, if you dock with less than 5 protein, I don't regard those papers. Because those people happen to be lazy. They are lazy. How will you do a docking with less than 5 protein? And if you even look at the protein they are using, all the proteins are just pointing to one mechanism. They will tell you, I want to discover a drug that will attack that mechanism. Yes, it's good. But we have polypharmacology, which explains that a disease has so many mechanisms of interaction. And these mechanisms are interconnected. So we can now develop a drug that can actually attack most of these mechanisms. That means we are developing a potential drug. Part of what we shall do in bioinformatics is to learn about protein preparation for molecular docking, how we can retrieve our protein and prepare the protein for docking. Then we shall understand protein domains, how to identify active sites. This is another overlooked area in drug discovery, where people find it difficult to identify the actual binding site of their target interaction. They just dock using blind docking approach. But it's very good we identify our active sites, because that will help us for validation. So we shall look at that. In overall, this module links disease biology with drug design. So we are giving you overview of what you're expecting in this training, and you can't afford to miss any day. We shall look at natural product mining. This is a little bit similar to phytoinformatics, but with a little different. This module teaches participants how to identify medicinal plants relevant to specific disease. So, okay, I want to work on ulcer. You don't just use any plant. Be able to teach you how to identify those medicinal plants that has a history with ulcer. We shall link ethnobotanical knowledge with computational screening. Of course, our backbone will be ethnobotanical reports, how it has been used in our various ethnic groups in our villages in treating those diseases. So we shall be going back to our village to get not just browsing and having data online, but a proof that this plant has been used for this disease. So our major focus will now be, if this plant has been used for this disease, which compound in that plant is responsible for that disease. We shall be able to see how we can select lead compounds from complex phytochemical profiles. How we can identify a lead from a library. Natural product mining will also help us to rationalize compound selection using biological relevance. So we'll not just learn how to select compounds, but we'll select compounds based on the disease of our interest. Very important. In overall, this module is valuable for pharmacognosy and natural product researchers, because which are basically open us to how we can mine our natural product and link them to our disease. Okay. We shall look at toxicological screening. I've taken my time to actually look at FDA standard for drug approval. And I noticed that in FDA, once you apply for a drug, the first thing they are looking at is not the efficacy of that drug, but the safety of the drug. So if you are to conduct toxicological screening in the lab, I don't think you have the money to do that. But with the help of these computational tools, we shall be able to learn in silico toxicity models. There are some models now that can predict toxicity. Though most people do not rely on that result. But when we get there, I will explain to you how those results are valid. Because once you introduce a result from computer, they will tell you this is computer generated result. They will say this is AI. They will tell you how will you rely on computational results. Those computational results, they were not just generated by the computer, but they were generated by algorithms as a result of experimental data that has been used to feed or to program that algorithm. So when we get to that part, I will clearly explain the validity of those results, which expose you to how you can be able to predict hepatotoxicity, mutagenicity, carcinogenicity, and cytotoxicity, and all the rest. So all these are what you're supposed to conduct in the lab, but we shall be able to see how we can screen those bioactive compounds based on toxicity. We shall be able to teach you how you can interpret toxicity endpoint, how you'll be able to interpret the toxicity results. There are about 15 toxicity endpoints which I'm going to expose us to, 15 different toxicity parameters that are essential for drug approval. Then we'll learn how to eliminate, very important, how to eliminate unsafe compounds before experimental testing. In fact, even before we start doing docking, we'll learn how to, if I have a library of 1,000 compounds from a plant, let's say I have 1,000 compounds from Moringa oleifera, we'll be able to learn how to filter out those compounds that are toxic. We'll screen them out and leave only the ones that are non-toxic. These are parts of the training under toxicological screening. Then at the end, this will help to reduce failure rates in later drug development stages. So the whole of this module is to help us reduce failure rates. Because you know that in drug discovery, we have a 90% failure rates or chances and just 10% success chance. So just bear in mind that if your goal is not just to end in paper, but to develop a product, you have a failure chance of 90% and just a 10% success chance. So by the time we bring in these screening tools, it will help reduce our failure rates. Then we shall look at drug likeness models. This model will focus on evaluating whether a compound is suitable as a drug candidate. It covers Lipinski rule of 5. It covers other likeness filters like Weber, Goals, and others. One thing is to have a compound, but another thing is, does this compound look like drug? Can it serve as drug? So we shall learn drug likeness models. The physiochemical properties analysis, those properties that helps those compounds to be suitable as a drug. We shall learn lead optimization principles. I discussed something on this area last week when we had a 2 days free workshop on macrophore-based drug design. I explained that one of the usefulness or application of macrophore-based drug design is for lead optimization. So we shall look at that, how we can make a compound to look like drug. Most times we notice that there are some compounds that have good binding affinity, but by the time you conduct your drug likeness test, they fail, maybe due to their molar weight. You may see compounds that are larger than with a mass of 500, with a mass bigger than 500 grams per mole. What do? We now look at lead optimization. It is either we reduce the size, but not just reducing the size, you must take note of a part that we are responsible for such interaction, then remove those parts that we are not actively involved in the interaction. Then we modify the drug. So we shall look at that aspect when we get to drug likeness models. In overall, participants will understand how to identify drug-like molecules from large compound sets. So another module we shall look at in this training is pharmacodynamics. Thank God for our friends from pharmacology department. When I was doing a course on pharmacology, I was introduced to, I was told that pharmacology has two branches, the pharmacodynamics and the pharmacokinetics. So we shall look at the pharmacodynamics, which is how those compounds interact with our body. The effects, their physiological effects, we shall look at them. So when we get to this module, it will expose us to molecular docking. We will look at the principles of molecular docking. We'll look at ligand and protein preparation. We'll learn about the docking protocol setup, interpretation of binding affinity scores, analysis of protein ligand interactions. So, in fact, we spend more time here because a lot of us who do docking don't know how to interpret the interaction. Most of us are interested in the binding affinity. But even though we generate 2D and 3D interactions, in some papers I see, they barely interpret that interaction. They only told you that there was a conventional hydrogen bond interaction with so amino acid, and this is the implication. That is not how to interpret a molecular interaction. So we shall be able to learn how to interpret a molecular interaction, the ligand protein interaction. At the end, the module will enable participants to predict the mechanism of action of those compounds at the molecular level. We are going to be learning a lot in this training. Finally, we shall look at the pharmacokinetics, which is how the body reacts with the drug. The drug that is entering into the system, these are herbal products. How will the body react with that drug? How will the body absorb the drug? This module introduces absorption, distribution, metabolism, excretion, and then predict their possible mechanisms. So it will predict their possible mechanisms. See how, in fact, in absorption, there are a lot of parameters we have to study in absorption. A lot of persons have been doing so many work on ADMET, but when I still look at these papers, I see great limitations. Because I love reading papers. I love reading papers because I want to see what people are doing. So we shall teach you how to interpret absorption. In absorption, there are about more than 5 parameters to tackle to the PGP. You're going to learn their implications. The HIA, you learn their implications. What the role they play in absorption, distribution, metabolism, and excretion. We can look at oral bioavailability prediction, and then the blood-brain barrier permeability, this vital point, the metabolic stability of the drug, the clearance and half-life estimation under excretion. In overall, participants will learn how to predict the behavior of drugs. So participants learn how to predict how a drug behaves inside the body before experimental studies. So by the time we are done with this with all this module, definitely you can be able to develop a prototype, even if you don't have the facilities to go for a large production, but at least develop a prototype that will be effective, then go for your wet lab analysis for validation. So far so good. I've taken my time to explain what we need in what will be covered in this training. So before I move further to the first module, because we have started. So before going to the first module, do we have a question? I'd love to take a question before we introduce our first module and we start. Hello? You can hear me, right?
Yes, we can hear you.
All right.
All right. Yes, we can hear you.
Yeah. I have exposed us to what we are expecting. So do you have a question? When you have a question, so in the absence of your question, we'll proceed to our first module. Okay, I get it. Okay, Mahmoud Lawa, your hand is up. Go ahead.
Good evening, sir.
Yeah, good evening.
Yeah. I wanted to ask, in this module, are we doing MD simulation, or is not related to Pharmacon, this internship?
Okay. Well, thank you for that question. MD is always the talk of the town. Everybody are now interested in doing MD. Most times, why we barely bring in MD is the issue we have in MD: high system performance. Most of us don't have that system that could be able to carry on MD. So if not, MD is not difficult. Just the running period, the number of nanoseconds that you allow it to run. If you begin to run MD with the kind of system some of us are having here, it will just crash your system. When I was doing MD, I know how many times I changed my system, like twice. Change hard disk, change processor, because of the number of times. I can't do MD with 100 nanoseconds. That is, I'm doing nothing. At least 200, 300, 500 nanoseconds. So the challenge is the system capacity of our participants. Now, MD is really necessary. MD is really necessary because it helps you to now identify the stability and the flexibility of your complex, your heat, or your lead. Because one thing is that you having a high binding result, another thing is that interaction stable. So MD help us to do that. Unless we can have people, because I can't teach MD when I know that the participant doesn't have the system to perform it. It's a waste of time. Like when I learned MD, I never had a good system. So I was just paying attention. I was not practicing. And we can't do such kind of training. We have participant doesn't practice, they just listen. Unless our participant has that confidence that, okay, we have good performance laptop, then we run MD. Otherwise, what we'll do is that if we have a job that we want to do for MD, we have people we collaborate with that runs our MD for us. We have people we collaborate with. Most of them are not Nigerians. I don't trust any Nigerian to run MD for me. Yeah, seriously. So that is not the challenge. So we will not include MD in this training.
So for all computer-aided drug discovery, there must be MD as the final this thing.
Yes, there must be
Mm.
MD. There must be MD. Now, the reason why we say there must be MD is after you have done your docking, although MD comes in very mostly when your target is to synthesize that compound. But when your target is to develop a herbal product, like that is my target, developing a herbal product, I don't need MD because I'm not going to throw away or modify any compound. I'm using them as they are naturally. I can't modify a compound in moringa leaf and put back that compound inside the moringa leaf. So in this Pharmacognosy, MD will not be necessary. Whenever your aim is to develop a herbal product, then MD will not be necessary. Where you just bring in MD is you want to know the stability and the flexibility. But
For a
I
layman
prefer,
like us, what is MD?
okay, MD is molecular dynamics. Molecular dynamics. The word dynamics means change. Change. The molecular change. As your molecule interacts with your protein target, what kind of changes are associated? Like if I have a compound, maybe for instance, I have a artemether for malaria and it interacts with my Plasmodium falciparum receptor, what kind of changing will occur when the drug is interacting with my receptor? Will the drug be stable at the binding site? Will it be flexible? What happens at a change? What happens at a change in time? What happens in change in concentration, change in pressure? What happens after a long period of time? Will it stay, or will the interaction be stable enough? That is what MD idea is all about. So it's a simulation too, just like docking. So I will always appreciate people to do MD, mostly when they are we want to synthesize that compound. It was before I usually call for synthesis, but I've seen that if we keep following synthesis, we will not achieve all what we want to achieve. Maybe it's our children that will take over from us, except to have the money. So the way now in drug is Pharmacognosy. The future of drugs now is Pharmacognosy. That's the future.
Thank you, sir.
Thank you. So Abiodun,
Yes, sir. Thank you, sir. We are so grateful, sir.
thank
So
you.
you've answered part of my questions, and
Okay.
I want to suggest, sir. So
Yeah.
under MD simulation,
Yeah.
probably there is chance if you can take us through like two or three of the parameters, the results, and what do they represent, and little interpretation aspect of it, like RMSD, RMSF, the
Yeah.
ROG, the hydrogen
Yeah.
stability. Please,
Yeah.
sir, we have it, sir.
Okay. I will do that at the end of the sessions, the end of the training. I
Thank
will
you, sir.
expose us to that. Yeah.
Thank
Although we can do cloud-based now. We can use Google Column for MD now. So I think I'll put up a training for that using Google Column.
All right, sir.
Cloud-based. Yeah, cloud-based. So you don't need to complain about your system capacity again.
Okay, all right, sir.
Yes. Cloud-based MD simulation. So I'll put up a training for that.
All right. Thank you very much, sir.
Yeah, thank you. You're welcome.
I would like
Yeah,
to know the
what's
time.
me Owolabi? I'm seeing your hand up. Rotimi Owolabi.
Yes, I raised my hand. I part of the suggestions I wanted to bring was the cloud-based computing, but you have addressed that
Yes.
as a solution to the limitation for our laptops. Then
Yes.
another question or clarification I wanted to ask for is your comment about the use of MD only for synthetic drugs, for phytochemicals as well, talking about their stability after interaction with their targets.
Yeah.
I wanted to clarify. You said that it's not necessary for phytochemicals. I want to confirm that.
Okay, let me explain well on that aspect. The reason why I myself, I don't necessarily spend time in MD if I'm running. Research is your objective. So if your objective is that I want to develop a herbal product, and in this herbal product I want to develop, I'm using the natural products in that plant. So if I'm using the whole powder now, so I've done interaction, I've interacted, and I have my complex. So if I want to run MD will have to tell me the stability of those my phytochemicals. Now, what if the result shows that they are not stable? Does that really relate that plant cannot be used for the treatment of that disease because of their lack of stability? So we cannot relate that, because the plant itself has abundant multiples of bioactive compounds, and some of them work by synergistic effects. But MD does not give room for that synergistic effect, because MD look at a complex. So if I said to follow my MD results, which proves that my interaction for my phytochemicals are not stable, it means I'm going to reject the plant I'm using. But that is not the case in reality, because from experience, I've done a lot of work on phytochemicals, and when I interact MD, most of them are not stable core MD results. But when we tried using those extracts doing the lab work, we still observe good results. So that is why I'm saying that if your aim is to just use your product the way it is as a plant material, so you don't need much of stress yourself on MD. Or if your idea is to identify a lead compound from that plant, that at the end you are going to isolate it or synthesize it, then follow up other processes, you must do MD. So that if that heat you identified happen not to be stable, you modify it and go back, you optimize and go back, or you change your bioactive compounds until you discover a lead that can be very stable. Then you proceed to your isolation or synthesis, which from the products you can do your wet lab. So that is just the reason why I don't encourage doing a MD when it comes to developing a natural product drug.
Hello, sir.
Yeah, wait to me. I don't know. Are we together?
Yes, we are together.
Okay.
I think you have clarified what was confusing. The fact that it depends on the objective of your
Yes.
If your objective is limited only to just bringing out the active component, fine. But if you want to develop active component to pure compound for drug purposes,
you must do it. You must.
Thank you. That's what I wanted to find out. Thank you.
Thank you so much. Okay, Dr. Vincent.
Yeah. The other thing I wanted to contribute is that
Yes, you may have to do MD simulations if your goal also is to publish the work.
Good. Yeah.
Yes, if you want to do some publications these days, they require MD simulations to make the publication very potent. So that may be one reason why some kind of study require MD simulations. Thank you very much.
Yeah. Thank you. Ayofe.
Okay, thank you, sir. Well
Thank you.
done, sir, for this beautiful presentation and the insightful presentation that you have actually opened our eyes to see the beauty of this pharmacognosy. What I wanted to add is what Dr. Vincent just actually impute now concerning is about the bureaucracy of publication. So it's actually about the bureaucracy of publication. Actually, if you do whatever you do, the end product of it, you are the owner of it. So if it works, just like you said that if you do MD or you do not do MD, you are going back to lab. I think if you do that MD, it will enrich the paper and you equally give you discuss the result. Though the MD simulation, there is no stability in MD simulation, but in vivo wise, there is reaction. There is the product actually work. So it's just a bureaucracy of publication. That is what I wanted to add, though Dr. Vincent has actually pointed it out. Thank you.
Yes. Okay. I forgot that we are 90. In fact, is it 90%? 100% academia. So sorry for that. And paper is what one. sorry for that. So I'll put up a training on MD using cloud-based computing. That would be very good for us, so that we can stop spending that money we pay people outside to do MD. I'll put up a training for that. Although we have a training one of these months on DFT, Homo and Lomo. So it's still part of what we need in drug design. We are going to look at in the next 1 hour. We just consumed first 1 hour. So in the next 1 hour, we shall look at the first module. So the training we hold for Tuesdays and Friday, as explained in our mails. So after each training, after each session, there will be our assignments. Assignment will be given to you at the end of the session. So you have time, because we made it flexible for a lot of us who are academic staffs. So it will not choke you up with your academic duties. So after Tuesday, you have Wednesday, you have Thursday. They will meet Friday again. You have Saturday, you have Sunday, you have Monday, you have Tuesday, you have Tuesday will come back. So we shall be giving you assignments to just ease up your burden and attempt what you can do. Then as our custom is, we love projects. We love projects in Hendeb. The criteria for this 1 month training is that before we give you your certificate, you must have a manuscript ready for publication. So it will not be bad if you put up a paper at the end of the training. The one will see that we cannot be able to give you a certificate. But for our free trainings, we do have mini projects because it's free training. So, but for our 1 month training, you must have a paper. So you must do a comprehensive work for that. So on that note, we shall start with the first database, the first module. Yeah, are you still putting up your hand? Your hand is still on. Yeah, I hope your hand is still up. Okay, all right. So we shall now begin with the first module, which is Phytoinformatics. Phytoinformatics. When we talk about phytoinformatics, what comes to our mind? You know that in pharmacognosy, our interest in pharmacognosy is how we can develop medicinal plants into medicine. So in phytoinformatics, it is all about studying, retrieving, storing, retrieving, manipulating informations that are related to a plant. Retrieving informations, storing informations that are related to a plant. And the kind of informations you are going to retrieve are chemical informations. When you want to study the chemical constituents in a plant, that kind of study is what we call phytoinformatics. And it is the foundation in pharmacognosy. Because if a plant, and I was talking to my project students today at the office, that if a plant, yeah, I'm not showing my slide again. I'm explaining. Yeah, I'm not showing slide. So if a plant can be used for a disease, then that plant must have some compounds responsible for the treatment of that disease. So the process of discovering the bioactive compounds in a plant, retrieving the bioactive compounds in a plant, is what we call phytoinformatics. So if we have a plant, let's say we have let me come in the angle of nutraceuticals. Let's say soybean. Now soybean is good. Soybean is good for children. Very good as part of their winning food. It's nutritious, is medicinal. So if soybean has been reported to possess anti-diabetic potential, so our job is to identify at least 1,000 bioactive compounds from soybean, because we don't know the actual bioactive compound that is responsible for that activity. So our job in phytoinformatics is to screen at least 1,000. When I look at some, I had a training last year around June. That was in a 3-month training. I told the participant to identify 1,000 bioactive compounds from their various plants, and they are shouting Jesus. They are mentioning Jesus. What does Jesus have to do with getting 1,000 bioactive compounds? If you cannot achieve 1,000 bioactive compounds in that your plant, you have not started. The reason why we must achieve up to 1,000 is a plant has thousands of bioactive compounds. A plant has multiple bioactive compounds, and we don't know the exact bioactive compounds. All what we know is that this plant is effective for this disease. So for you to screen that's your plant, we must achieve 1,000, at least 1,000. So the question is, how can we achieve 1,000 bioactive compounds? 8 years ago, about 8 or 5 years ago, when we started this job, my first research work on the Cromolena odorata in our institute, we mined 1,500 bioactive compounds from Awolowo. That is a Cromolena odorata. It will be stressful, but that is what we are going to do. That will be our first assignment. And you're not doing it for me, you do it for yourself, for the sake of your research. So you want to formulate a drug, you want to build a herbal formulation with moringa, you want to build a herbal formulation with bitter leaf. then let us screen the bioactive compounds. So how do we achieve that? That is the question. How do we achieve this phytochemical screening, mining a large number of compounds? I will give us 3 ways we can achieve that. 3 ways. 3 ways we can achieve that when it comes to phytoinformatics. One of them will depend on your finance, your pocket. So the first approach for phytoinformatics is your wet lab. Your wet lab. You are going to do perform chemical characterization. You are going to perform chemical characterization, not with your normal one solvent. When I see some papers, you see some persons who will just use ethanol and extract and do their work. At the end, they will claim that they have gotten a potential drug candidate. That is rubbish. And if you look at the GCMS results from the ethanol extract, they mainly end up achieving like 50 compounds. Meanwhile, the plant had more than 50 compounds. Even there's a probability that the 50 compounds they got in the ethanol extract may not even be among the ones responsible for that physiological activity. So one of the ways we are going to do that is you perform your wet lab, your characterization. Use it at least 10 solvents of wide polarity, wide range of polarity. Because the chemistry of extraction is based on solvent lightness. That those solutes, the bioactive compounds that are light with your solvent will be dissolved, will be extracted. So if you are using aqueous extracts, then you will end up only having those bioactives that are soluble in water. Then you look out for at least 10 solvents. 10 solvents with wide polarity. Maybe 5 polar, but the polarity is different, and 5 non-polar with wide polarity range. Conduct your extraction for each of them. You end up, even though some compounds might be common in some of the extracts, but you extract all of them and then do your characterization. So the first approach is your chemical characterization. Then in your characterization, what kind of characterization I don't know do? How we prefer you work with 2 GCMS, that is one for gas chromatography and one for liquid chromatography. So you perform your gas chromatography using GCMS, then you perform your liquid chromatography using HPLC. Reason because your GCMS will only give you the result of volatile compounds, while the HPLC will give you result of non-volatile compounds. Some of them are peptides, so we still need them. So you do a lot of work. That means if you have 10 extracts, you take those extracts for GCMS and HPLC, GCMS and HPLC, GCMS and HPLC. By the time you combine these results, you begin to imagine the number of bioactive compounds you are accumulating just when you are doing your wet lab. Then the other approach, but then if in case you are combining, you are doing a polyherbal mixture, then you need to prepare your formulation from the plant you want to study, and then when you have prepared the formulation, you use the extract of the prepared formulation for characterization. But when your interest is just on the unique plants, then use that plant for the extraction. But if you want to prepare a polyherbal formulation, of course, in herbal product development, we don't use one single plant. Now, if I want to do a work on, I want to develop a herbal product for maybe ulcer or for diabetes, I make sure that there will be a plant that is responsible for, that has the potential for the diabetes. Then there must be a plant for a plant that has a potential for inflammation, and the one that has a potential for oxidation, an antioxidant and anti-inflammatory. So these parts must be there. If you're developing a herbal product, there must be an anti-inflammatory agent, there must be an anti-oxidative agent. Most importantly, these two, at least, most importantly, two must be there as part of your ingredient. So if you want to do a polyherbal formulation, then you first prepare that formulation and then use formulation for extraction. So you are going to do each of them, but the challenge you have now is the money to perform that kind of multiple extraction, the money to purchase this solvent. So you don't work alone now, you have a team. I was lamenting on the issue of when you hear collaboration, what we really do. I made a post on my LinkedIn. When we hear about collaboration, what comes to our mind is for paper, and that is a madness. So the essence of a collaboration should be in research, not just in having papers. So you cannot collaborate with some people who have this money, get the solvent, perform your extractions, and then do your characterization. So you combine your results. You are building your library. Then the second approach is you can leverage on what some persons have done. You can leverage on what some persons have done. You look at for some of these reputable journals, look at their characterization, because there was a time when characterization was the top notch in research. If you have not done it, you have not done any work. But I think now nobody praises any work on characterization. So a lot of work has been done already in the past for that your plant. So look out for them. Retrieve them as much as possible, at least 500. So even if you don't have the money, for the case for the purpose of the training, you may not be able to achieve the wet lab, so you can leave that. Then you now focus on assessing other publications, literature reviews, mine some literatures, get their bioactive compounds as much as at least 50. This was why I told those in Genomic Institute that you need 50, at least 50 bioactive compounds that has to do with chemical information of that your plant. They are available, they are everywhere. You just have to stress yourself and retrieve them, up to 50. So by the time you combine their results in your database, in your own library, combine, download them, get their results. If join with the one you did in wet lab, if you're able to do that, you will see that definitely you are building a library, very large library. And then you can now get exposed to phytochemical database. So that is what we are going to look at now. We're not going to expose you to some of the phytochemical database. Unfortunately, thank God, most of us here are Nigerian. I hope we know that Nigeria does not have a phytochemical database. I don't know if you are aware of that. Are we aware?
We are aware of that.
And something needs to be done. Though in my institute, we have started a work since 2 years on that. We have done a lot of work on that. There's one I saw, but some of us who are in academies, I don't know, we like money too much. We pursue grants. Some of us pursue grants and we don't use it for the purpose. Is where shall let me not go into that area. That is not what I'm here to do. I saw one on I don't know. Yeah, most of us must have even come across it. NMPPDB. Yeah. So let me start with our Nigerian phytochemical database. This is the only database I was able to lay my hands on. If you're ready to work with us on this, we are open. We are doing a work for our fact. We have gone far. We have tested the database that we propose, and is superb, because we lack documentation here in Nigeria. We lack documentation, and is a call to our pharmacognosy departments. They can develop their own database for themselves. So I came across this database last year. Is very Nigerian logo, Nigerian medicinal plant phytochemical database. If you look at it, it is dry. They did that because of the TED Fund NRF grant. So they collected. Sorry if those persons who did that are here. Apology. Apology, please. But you are giving 50 million or close to 50 million to do something. At least come up with something good. So if you look at this, is really, you can take your time to look at it. So these are part of those database we are going to mine, but we shall look at impacts. Most of us are familiar with impacts, so we'll start with impacts. You can now look out for some database. There are some database where informations of your plant has been deposited. One of those databases is IMPACT, Indian Medicinal Plant Phytochemistry and Therapeutics. So what you do is you come to this, I will expose you to about 5 of those databases and how to use them. You gather the bioactive compounds from these databases. So by the time you join your wet lab, you join your literature mining, you join the one you achieve from this database, don't you think you may achieve up to 1,000 or more phytochemicals? Then you screen them. You identify the ones that are toxic. You see if a good number of the bioactive compounds are toxic. So that will enable you to know that if I'm using this plant, this plant has a lot of bioactive compounds that are toxic, or the toxicity is there but is minimal, because we are using the whole plant powder. Although we'll come to a little bit on, is it ideal to work with extracts in the herbal formulation or to work with the powder? Some persons still have that issues. So impact is one of the database we are going to explore right now. How you are going to do that is you just search for your plant and you download all their bioactive compounds. So that will be our first assignment today, to download 50 literatures and retrieve them and then use your impacts. I'm sorry if that will be too big for us, but that is the essence of the training. So impact has about 4,010 Indian medicinal plants with about 17,967 phytochemicals. Now, some of these plants are also found in Nigeria, so we can mine them. Although we know that there will be diversity. Yeah, there will be diversity because even the ones you plant, like take for instance, if I cultivate pawpaw, or if I grow moringa in Imo State and grow moringa in Adamawa, if I characterize them, even their genomes, if you characterize them, there will be some diversity. So we still know that some of these plants we are sourcing from this database, the phytochemicals there might not also be same with the one we are going to use in Nigeria, but there will be little difference. So I will encourage you to get all the bioactive compounds there. So, and there are some plants you may like to work on. You will now notice that those plants are not found in some of these databases. So it means I don't want to spend time by yourself or spend your money in doing your characterization. So how do I use our impact? You can search based on phytochemical, or you can search based on therapeutic use. So, but we shall look at for today searching based on phytochemical. Hope you can see my screen.
We cannot really. Yes, sir.
Not clear, not clear at all, sir. I can't see the screen, and I wanted to ask that the impact, the spelling, so for us to follow very well. Sorry.
Okay, but can some person see my screen?
Yes, I can see it from my own end here.
Yes,
Some
I
of
can
us,
see it.
some of us can see it clearly.
Yes, I can see.
Okay, so that means that will be network challenge for, yes, at their own end. So the impacts, I want to drop it now at the chatbot. So
IMPP80.
Yeah, IMPP80. So no need to just type it. IMPP80. All right. So if you are searching by phytochemical, choose an Indian medicinal plant. So you click here, and then
The link is covered the search icon. So
no, just type IMPP80.
yes, when we get to that website. So okay, are we clicking on browse?
Yes, when you open your website, as if you want to browse, type impact IMPP80 database and send.
So I have different icon.
Pick the first one, the first one.
Which is home.
Is
it home or browse or basic search?
Oh, I am getting a
little browse, basic search, advanced search, statistics.
Okay. Are the impacts? Oh, sorry. Oh, all right. I got what you're saying now. Just scroll down a bit. You will see browse. Okay, or click on browse. Yeah, better. Click on browse, or you scroll down a bit. You will see the browse showing phytochemical associations. All right. Are we together now?
Yes, sir.
All right, thank you. So we are going to search by phytochemicals, because what we want to do is we want to retrieve some of those bioactive compounds that are associated with our plants. So what you just need to do is you can choose by the plant, you can choose by phytochemical by searching a phytochemical, but I would prefer we work with choose the plant. So click on this drop down, and the list of the plants will pop out. Then you can search the plants you want to work on. So there are about 4,010 medicinal plants already arranged in alphabetical order. So which plant do you want to work on? Let's say I want to work on the one I worked on before, Comolena odorata. You just type Comolena and you can select. It's already showing here, Cumulena odorata. So I can just search for it. So this is my Cumulena odorata. So you search and click on it. This part usually is the most stressful part when it comes to Pharmacognosy. Is, to me, is the most stressful and annoying part, but we must do it. Except you're not ready for the training, but please, I know we are busy, we have a lot of professors, but just try your best. You can do it. Now, if you look at this platform now, at my right, where my cursor is a cluster showing the bioactive compounds, the range of the bioactive compounds. So you see that traditionally there are much bioactive compounds. If you scroll down, you can see how many of them here showing. If you scroll down, you see showing 1 to 10 of 282 entries. So there are 282 entries here for Cumulena odorata. There are 282 bioactive compounds. Now, when you are mining the bioactive compounds, when you are trying to retrieve this data, please take note of the plant parts. You will take note of the plant parts. So let's say you want to retrieve chemical informations of this Cumulena odorata. That means you have to create different folder for the leaves, create a folder for the stem, for the flower, for the roots, so that you don't end up mixing up the bioactive compounds. So that we can know that, okay, if we are working with the flower, okay, these are the flower parts, these are the compounds found in the flower. If we are working with the leaves, so these are the compounds found in the leaf. And the people that built this database, they also took notes of this major point. You will see that their compounds were arranged according to the plant parts. So these ones are area parts, and this is the names of the compound. Let me increase the.
Sorry, we are having challenges typing inside the choose a plant. So because
Okay.
it's not stable.
Yes, can you click on that drop down?
Okay.
That's a little arrow.
Okay.
You can as well scroll down.
Yes, once you click it will bring down the list, so you can scroll down to the alphabet range of the plant you want to select and check if the plant is there. Most times the plant of your interest may not also be found in that database.
Not here. It's not here.
What's the challenge? What's the challenge?
I can't see chromo. I can't see. Okay, CH. Okay.
Is there?
Look for CHRO. You will see there. It's there.
Now from there, I clear my own.
CHROMOLAE.
I've also seen it too.
NA.
Odorata.
Odorata, yes.
All right. So if you look at, you see that it was arranged based on the plant part. So all these bioactive compounds are the ones from the plant part. And one thing I love them is that they referenced the publications. Some of them were not just gotten from publications, some of them were also gotten from textbooks.
Sorry, I want to ask, in case if you do not see the plants you are looking for, so is there any other thing you can do about it? In case if you do not see.
Good. There are other database which I will also introduce. We have Dr. Dukes. We have Dr. Deuce owned by American. So you can also check. I will give you, I'll introduce us to 5 plant-based database. Plant database. So if your compound, if your plant was off among the ones in the MPAT, then you check in the other database that we expose it to. But even if your compound was found in impact, also check other database and download them as well. So you see that you are doing a very big work here. So you must make use of the 5 or as much as possible that I'm going to give to you. You must make use of them. You must make use of them. So if your plant happens to be found in all, glory be to God. Then if your plant was not able to be found in all, fine also. I was privileged to supervise some students at Anambra State University on Bioinformatics. When I told them to download from 5 databases, some of them had to say, sir, out of these 5, my plant is only found in 1. Meanwhile, those plants we are found in all of them. But because they want to ease up the stress. So, but I will encourage you to take up the stress. It will only take your time. Because some of these database, in fact, all of them does not have automatic download. So you are going to download them one by one. They don't have automatic download, like where you just download all and all the compounds will download. You have to download them one after the other. So you see the stress in silico drug discovery. I know that one could say, why can't I just use my AI and type list of bioactive compounds in Cumulina water, so AI will pop out the bioactive compounds. I don't encourage that to, because your work.
What about if you use coconut database? Won't you be able to download all?
Coconut. Can coconut select plants? I've used that before. Can coconut identify different plants? If coconut can download all, fine, that is good. Yes, if it can download all, that is good. But coconut for identifying plants, let's see, Cumulina odorata, but I don't think so, because I've used that before. Let's see. Let me see what you just asked, if it's possible. It's a good question. Okay. Until it's still searching, but I don't think so. No result found. You can try coconut, but I don't think so. If coconut can give you a bioactive compounds for a plant, but we figure it out. Yeah.
So what if you have like a compound now, and okay, you found it in impact, but you said you give us up to 5 databases.
Yes.
If I can find my plants with with the bioactive compounds in one database, what's the use? Do I need to start looking for them in different databases also again?
Yes, so you must. The reason why you must is, oftentimes some of the reported bioactive compounds in database 1 might also be found in database 2, but not all. So you are going to get all, then remove duplicates. In fact, while you are downloading, your system will even tell you that this one has been downloaded before, meaning that it has been gotten before from one database.
Yeah.
Yes. You get that?
Yeah, I do, sir. I do, sir.
It will be stressful. It's
Sir,
only time to take you.
sir, I have a question, sir.
Yeah, go ahead.
There's something you said about the plant. You were saying that if you want to work with, like you work with different part of the plant, if it is the leaf, if it is the stem, the root, then what if I decided to work on maybe on the leaf alone? Then forget about to know the active compound found in the leaf and not bothering looking at the root and what if my area of interest is on the leaf?
Okay, that means
focus on the leaf.
Yeah, yes. That means if you are looking at for this database, if you are using this database, since the database classifies the bioactive compounds according to the plant part, so you just focus on the one for the leaves and download those ones. And then if you are using literature mining, you look out for literatures that report only on the leaf and pick those ones, irrespective of the solvent used.
Okay, sir.
But
sorry, how do we separate our search?
How do you do what?
How do we separate our search to leaf, roots, and some other things?
Okay, it just if you are downloading, I'll show you how to download. But if now that since I opened my impact and the first thing I'm seeing is area parts, so after downloading for area parts, in your download, you just have to copy your downloads, create a folder, and push them into that folder. Name it area part of Humulina odorata. So then when you scroll down again to other parts, to avoid mix up, when you scroll down, I'm coming. Now I'm seeing flower. So I will also remember I've cut the ones I've downloaded for area parts. So there are no more in my download. I've cut them and paste in a folder named area part of Humulina odorata. Then I will start downloading the ones for the flower. So after downloading the ones for flower, I will also do the same thing. So if I open another database or journal that has area parts or that has flower, I will download them, cut them, and join with the previous folder I've created for flower. So you will create the folder yourself. Is that clear?
So how do we download?
I don't understand. Please, I don't understand.
Okay, now let me show you something now. Let me show you.
Okay, sir.
Yeah. So how do we download it? That is most importantly. Since we have this already, these compounds have their identifier from impact. But for the sake of uniformity in our research, we are going to download them from PubChem. So we are going to download them from PubChem. So what I'll just do is to open another browser, open PubChem, and just type PubChem. PubChem is another database. So not really a phytochemical database, it's a chemical database. So I will explain more about PubChem when we get to chemoinformatics. But we shall be using it to download the compounds. That will be very easier for you. So what I'll just do is to remove my PubChem from here so that it will be separate. Then I'm going to minimize this. So this will be easier for me, very easy for me to be downloading. So you can open a separate window, and for your PubChem, then you have another web page for your plants. So what you will do is this PubChem is where I'll be using to download my compounds. I can equally download from here. If I want to download from here, I will just click on this compound. If you click, it will take you to the profile, the platform for that compound. Then if you scroll down, you will see download. You see download. You can choose any of the download option you want to download. You can pick, usually pick 3D, and I advise you to take 3D SDF. But because of the stress going back to and fro, after clicking on the first one, you will now go back and click on the second one, then you come back again. And because of that stress, number one, and number two, if I download from here, it will download with the identifier. So it will not download with the name, it will download with the identifier. IMPHY. So this is the code for it. Now, if I use another database too to download, it will also download with their own identifier. So, but for the sake of uniformity, so we'll be using PubChem. And by the time I have shared my screen into two, it will just be easier for me to copy from here. So you copy from here, you go to this PubChem page, you paste, and then you search. You search. So when you search, now before you download from PubChem, because now we want to retrieve their structure, we want to download their structure. Most importantly, look at this MF. This MF represents the molecular formula. Now, some of these docking tools, like the one we shall be using, Pyrus, does not recognize silicon, Si. So a compound that has silicon, if your docking protocol is based on auto.vina, it does not recognize Si, silicon. So if you have a compound that has silicon as part of your bioactive compounds, it will interrupt your docking process. So once you look at the MF, you check if there is Si. If there's Si, leave that compound, just leave it, don't download that compound. But if you look at it, you just have a carbon and hydrogen, then you go ahead. So you click on this compound CID. Once you click on the compound CID, then you click on download. There are a lot of information here, but that is not before us.
Hello, sir. How do you do the copy, sir?
Okay.
Because when I wanted to copy, it was giving me link.
Okay, no, you are clicking on it. Just can you see my cursor? Can you see my cursor?
Yes,
so
sir.
hold your left mouse and drag the cursor.
Okay.
So you can keep the cursor outside first, close to the end or the beginning, whichever case. Then hold your left mouse and drag it to cover the chemical name.
Okay, sir.
So if you have done that, you copy. Hope you are working with me. Like, yeah, do what I'm doing.
Yes, sir. Yes, sir.
Because I believe I'm not much fast, because I want you to follow up.
Not at all. You are not fast.
So when we have the whole of this, the whole of now till next month beginning, so we have a lot to do. So when you copy, you come to this white space and then you paste. When you paste, you hit your enter key. So as you hit the enter key, so what I'm saying is the Pyrus we are using does not recognize silicon. Now the problem is you must be careful, because if you are docking 100 compounds at a time in your Pyrus, and the one that has silicon is the 100, is the last one, or the 99 compound. After docking up to 91 or 90, or the one that has silicon, it will show you error. And once it shows error, all the time you spent in dotting from one to the one that has silicon is just a waste. So you can't retrieve the result for those ones that have been docked. So that's why you be very careful while you are downloading the bioactive compounds. Don't pick the ones that have silicon, because silicon is not recognized in auto.vida. But if you are using other tools like MOE, most times if we are doing training like this, we use open source tools that one can easily access. Although we are planning of organizing a professional training for 3 months, and that is where we'll bring in that MD. So we shall now give you access to some of these licensed tools like Mastro, Strundiga, mostly MOE. They are very good. So they can accept any kind of structure, kind of atom you want to input. But some of these open access, if you look at, not that piracy is bad, piracy is good. It's even one of the, from statistics, is the most commonly used dotting algorithm, dotting tools. But it does not recognize silicon. It does not recognize silicon. So you look at this MF and check it as SI. If there is, leave it. Don't stress yourself. But if there's no silicon, then click on this compound CID number. Once you click on it, it will open up the compound. Once that compound opens, now you click on download. Click on download. So once you click on download, so are we on the same page?
Please come again. Please come again, sir.
At which point?
At where you are showing us the MF, sir.
So I can see your slide again.
Yes.
Okay, so what happened to the MF now?
Just continue from that side. It's okay. I can see.
Okay, can you see my screen? All right. So the next thing you will do after checking the MF that there is no SI, click on compound CID, the code, that compound CID number. Now, there is no need for you to take note of those compound CID. There's no need for that. Don't bother yourself trying to copy the name of the compound and the CID. Because when this compound will be downloading, it will download with the CID. It will not download with the name of the compound. So you don't even know which compound is that. We don't need to know. It is after we have done our docking simulations, when we have done our docking, the ones that happens to be to have a better interactions is now our interest. So for now, we don't have interest knowing the names of the compound. We'll just download them like that. So you click on this CID number 1060783. Once you click on it, it will open up download. Can you see the screen?
Yes.
All right.
Yes, sir. We can see.
So the next thing you will do now is to click on download. You click on download. All right. So are we on the same page now?
Yes, sir.
Yes, sir.
Thank you. So on this download, we are interested in the 3D conformer, but most at times not really most at times, a few times, sometimes you may end up seeing that the compound you want to download does not have a 3D conformer. It will end up having just 2D. No problem, you still download it. But if you are able to find 3D conformer, then click on save, this first one, beside SDF. Because you want to retrieve the structural data file. That is the SDF, structural data file. So you click on SDF. Now, those compounds without 3D that just have 2D structure, we shall learn when we get to Bioinformatics, we shall learn how to convert those 2D structures to 3D structures. So once you just click on save, that compound will download automatically. So it has downloaded with the compound CID. So now this compound now is for area parts. So since I've done that, I will go back for another one. So I will come for the other one under area parts. I will copy it. Then for you to go back, you click on this capital C here, a capital C, or this search icon. It will now take you back to where you can do your search. So you paste. As you paste, you hit your enter. So you keep doing the same thing repeatedly. You pick the first hit, the first best compound match. So you keep repeating what we have just done. You check the MF. There is no SI. So you click on the compound CID. It's something the only thing is that it will take your time. To just take your time. Then you click on download, and then click on, it has 3D, so SDF, you click on save, it will download. So let's now assume that we have downloaded these two compounds. These two compounds now has been downloaded, and they are under area parts. They're under area part. So I want to now pick the ones for flower. So since I've downloaded these two and they are under my area part, you just have to cut them. You just have to cut them, select them, cut them, Ctrl S, cut, create a folder, create a new folder. You just create a new folder. And you can name the folder area part. Comolena or C odorata area part. So, and then paste them inside the area part. So why you must cut them off is so that it will you not mistake them with the new one you want to download. So then I want to now download for flower, for the flower part. So you come back. All right. Are we still together?
Yes, sir.
So now, I want to do for the flower. Maybe I want to, I've done all the area parts. So I want to do it for the flower. So you scroll, you see the flower. So the flower, you can just myotic acid, you pick myotic acid, you copy, you come back to this place and you paste. So you paste, you continue. It's a stressful job, but if you can do that, it will be good for you, because you have just built your own working directory for the plant. So anytime you want to do docking with that plant, you don't need to start downloading again. You already have your license. So you can stress yourself once and for all. You check there is no silicon, you proceed. So we have 2 minutes to go. Yeah, I don't want to. You download. So when you download it, you choose a 3D conformer SDF, and you save. So now that we have saved, so assuming we have done much of the flower, then the same thing you have done to create another folder. You cut the ones all the ones you have downloaded from the flower, you cut it and create another folder. You create another folder. You name the folder C odorata flower. Then you paste what you have the cuts. You paste it inside the flower part. So this will enable you to be able to separate the compounds that belong to the flower and to the other part. So even if you are using literatures, maybe let's say from literature, maybe we did from the literature, let's say we said so if you are doing literature, you can look at for some search engines, which are PubMed, Scopus, you search in Google Scholar and all the rest. So I think it's still what we're going to be doing. But let me quickly say I have chemical characterization of chromolena. So if I'm interested in the leaf now, so of chromolena leaf.
They can't see your screen.
That will be network from your end. Anyone see my screen?
Yes, sir.
Yes, you can see.
Okay. All right. So I just searched chemical characterization of Chromolena leaf extracts. Chromolena odorata leaf extract. So I want to specify for the leaf. I want to specifically search for the leaves. So you can, irrespective of the extracts, yes, because we're not looking at based on extracts, we're looking at the compounds found in that plant part. So you can open as much as possible, download them. So when you download them, the same thing you have done too. You scroll down. This is chemical composition of the ethanol extract of water leaves. So let's see if there is a report on the bioactive compounds. Okay, there is a report, and since it's for the leaf too, you copy cryophyllene. You copy. You come to your pop game. The same thing. You repeat them over and over again. So that is what you can do. So it's not a big deal. Is it a big deal? I believe it's not a big deal.
Somehow, sir.
Not
Somehow.
So as we meet next, we meet again. No, not at all. Not at all. There is no somehow. It's just time. It's just time. But we have the time. You see, when we keep saying we don't have time, we have the time. We have the time. We have the time. I was discussing with my student today, and they said, sir, medically we should be sleeping for 8 hours per day. So once they mentioned sleeping 8 hours per day, I had to calculate if we sleep 8 hours per day, if we sleep 8 hours per day, in 7 days, we will sleep for 8 times 7. That is 56 hours. And that 56 hours is 2 days, 8 hours. That means in 7 days, you have consumed approximately 2 days and a half sleeping. Out of 7 days, 2 days and a half has been consumed by sleep. So the remaining 4 days and a half, the remaining 4 days and a half, probably one full day must have been taken for chatting and for relaxation and all that. So how many days do you have now to work? So that ideology is not there. Somebody asked me, okay, a person is here, but I will not mention. He said, do you sleep? I say yes, but I sleep 24 hours per day. And it's medically right. So I will encourage us. But it's your business, sha. If like you want to be sleeping for 8 hours per day, now you sabi that one. Just be eating good food. Be eating good food and be taking fruits. Then do exercise. If possible, we should having have 48 hours per day, not that 24 hours. So thank you so much. Somebody has reminded us that this is 10 o'clock because some person that have other tasks to do. Yes, I don't intend exceeding above 10 o'clock. So thank you so much.
Please repeat the assignment again.
Okay, the assignment is this. For now, don't enter into database because we are not done with the database. So work based on literature mining now. The assignment is mine up to 50 literature and load up to 50 literatures. And when you
repeat.
I will still paste it in our platform, but let me say it here. Then between 50 literatures, select any plant of your choice, any plant you want to work on. Between 50 literatures for those plants, for the plant you want to work on, use one plant. Download 50 journals that has to do with chemical characterization of the plant. Then download those, use your pumpkin, download their chemical constituents based on the plant part. Now, you are not limited to any plant part, as many as possible you can get from the different plant parts. So, don't say my focus is on leaf, not your own personal research. For the purpose of the assignment, we are working on the available plant parts you can lay hands on. If you claim that you only found on leaf, no problem, fine. But if you can see others, I appreciate. Then download them in public and group them according to the plant part. So probably if you pick this first journal and you downloaded on leaves, and then you saw another journal that has to do with the stem, download. Then if you see another one too that report on the leaves, the way you download, copy it and join the folder you've already created for leaves. It's a simple one. So for now, we are working with your literature mining. The assignment will be submitted on or before Friday morning. On or before 12 p.m. on Friday. Can we achieve it? Can we achieve it? I want you to stay your mind though. We can,
Yeah,
right?
it's achievable. What's the mode of submission?
Yeah, it will be 2 mail. I will drop the guideline tonight. Yeah. So by tomorrow morning, the video will be updated. So check your drive tomorrow morning and download the video for this session and use that for your personal time. Once again, thank you for being available. Thank you for coming around. We really appreciate.
Thank you. You are well done, sir.
We
We
really
appreciate
appreciate.
Thank you, sir. Thank you, sir. We're so grateful. Thank you, sir.
Yeah, thank you.
Sir, please, sorry, I didn't get the second part of that of the assignment. You said the first part, download 50 literatures, like you choose a certain plant, the chemical characterization of that plant, and then the second part, I didn't get it.
So download those compounds from your literatures. Download the compound using PubKey according to your plant part. Yeah, I will still drop the guideline clearly tonight. So I'm wishing you a very good night rest and a good sweet dream. I endeavor to read the textbook we sent to all those. I don't know if you have opened the textbook we sent to your Google Drive.
Yes, sir.
Very wonderful textbook. We sent 2 textbooks, one of Pharmacognosy, one on in silico. So just try and open those textbooks. The reason why we are doing that is so that you can get familiarized with what we are teaching. So yeah, you just try your best. If you need more, come to us. We'll dash you more.
All right, sir.
We'll dash you more. I want to appreciate once again the school that has the highest number of population here, those from University of Benin. Thank you for turning up.
Wow.
Yeah. Thank you. I really appreciate Professor Vincent for work done. Then on Saturday, prepare for our webinar on Saturday. We have a webinar on Saturday, free webinar, Saturday by this time, Saturday, 8 p.m. It will be so great. Let's discuss on Saturday. We are coming to discuss on Saturday. Nobody is doing teaching. We are discussing on feature-based drug design for biomedical and environmental sciences. The advert is on, register, is free. Thank you so much, and God bless you. Good night.
Thank you, sir.
Yeah, thank you. Good night. Thank you. Thank you so much. Thank you.